Alright, we're going to do the liver and enzyme lab. Now, every cell in your body produces peroxide when it goes through chemical reactions. And unfortunately, peroxide is poisonous. So what your body needs to do is have a way to break down the peroxide. And one of those things is catalase. Catalase is made by every cell in your body. And when it breaks down peroxide, it turns it into water and oxygen. So what we're going to do is we're going to systematically prove that the liver, which contains the enzymes, is going to break down the peroxide into both water and oxygen. So we're going to start with test tube number 1. It's going to be our control. What we're going to do here in test tube number 1 is add water. Water is usually what we use for controls. We're going to put that up to about a 2 centimeter mark. And then we're going to add a piece of liver. So the liver's going to go in, and we'll push that down. And you'll notice once it goes down in there, that no reaction took place. Surprise, surprise. So what was missing? There was something missing from that reaction. All right, now we're going to work on the second tube. The second tube, we're going to put peroxide in there along with the liver. So we're going to fill, just like we did with the first one, we're going to fill this one up to the 2 centimeter mark, but this time it's going to be with the peroxide. Remember, peroxide is going to be our substrate. And we're going to add a piece of liver to that tube, and we'll push it down. It gets down in there. Uncooperative piece of liver. There it goes. And notice this time there is a reaction. But now the question is, do we know for a fact that the peroxide is being turned into water and oxygen? We're not quite sure what those bubbles are. So that's actually what we'll prove later. So as these bubbles rise, the liver enzymes are turning the peroxide into, hopefully, water and oxygen. Alright, so the third test tube, we're actually just going to put peroxide in there and no liver, because we want to prove something isn't going to happen. So we're going to go ahead and put peroxide in the third tube, again filling it up to about the 2 centimeter mark. And I'm going to light a wooden splint on fire. And the technique we're going to use is we're going to let the wooden splint burn for a while, and then after a while, as it's burning, see how it's glowing red? What we're going to do is we would slowly put that down into the tube, and nothing happens. It's because peroxide itself isn't flammable. So we wanted to prove in this tube that peroxide isn't flammable, so that what happens next will be pretty cool. Alright, so the fourth test tube, we're going to put all the components together. We're going to have our liver, which has the enzyme. We're going to have the peroxide, which is the substrate. And this time around, we're going to prove that oxygen is what's being given off in this reaction. So here we go. I'm going to fill up the fourth test tube, again about 2 centimeters or so, with the peroxide. I'm going to add the liver. And push it down in there. And of course the same reaction is occurring again, only this time we're going to actually prove that oxygen is what's being made. I don't know if you know this, but pure oxygen in the presence of a spark creates a fire. So I'm going to do the same technique again. We're going to let that burn for a little bit. So now it's glowing. Oh! So it just keeps. Oh, and we burned it out, so we'll light it again. Now, the reason why it went out is because it's also giving off water. So water is in those bubbles as well, so sometimes we'll actually put the flame out. And let it burn for a little bit. So there you go. You can see that oxygen is being given off. The fifth test tube, we've actually been boiling. We put some water in a test tube, we put the liver in, and it's been boiling for a while. Now, if you recall from the notes in the lecture that enzymes in the presence of heat, if it's too hot, the enzymes will denature. That means the enzymes are going to change shape, and therefore the substrate's not going to match up to it anymore. So therefore the enzymes won't work. So we're going to prove that here. If you come on over, I've got water and a boiled liver in this last test tube. I'm going to go ahead and put some peroxide in. Now recall from the previous couple of test tubes that the minute peroxide and liver got together, it created a whole bunch of bubbles. This time around, there's nothing going on. So even if I did light a wooden splint and try to put in there, it's not going to react because no oxygen is being given off. So let's recap real quick our 5 test tubes. The first one was a control. We were missing something from that one. We had the liver and we had water, but we were still missing something. The second tube, we had both the peroxide and the liver, and a reaction did occur. We did see some bubbles in there. The third one, we lit the wooden splint into the test tube of peroxide, and we found out that peroxide is not flammable, so that when we did the fourth test, we knew that during the reaction that oxygen was being given off because it did reignite the stick, the lighted splint. And then our fifth test tube had the boiled liver in it. The enzymes didn't work, and we proved that because when we added the peroxide after it had been boiled, there was no bubbling.